double helix structure. Every side is made out of monomers called nucleotides. These nucleotides are made up of nitrogen, sugar, and phosphate groups. Purine nucleotides have double ring structure, while pyrimidine nucleotides are single ring structures. These structures are bases, which make up DNA. There are 4 bases, 2 single-ringed and 2 double-ringed. Single-ringed ones are thymine and cytosine. The double-ringed ones are adenine and guanine. A specific single ring pairs with a specific double ring structure. Adenine always pairs up with thymine, and guanine always pairs up with cytosine. The order of each of these bases will determine the genetic code or instructions they give. These instructions tell the cells how to make protein that the human body needs. In each cell, there is something called the nucleus. The nucleus has chromosomes or long strands of DNA. These long strands of DNA have sections of genes. These genes are the instructions which make RNA, and RNA is what helps make proteins. Now, the first step to creating proteins is transcription. This is when RNA is formed by using the DNA as a template. Unlike DNA, which is a double helix structure, RNA is only one. There are 3 major types of RNA. There is tRNA, mRNA, and rRNA. The transcriptions occur in these steps. First, an enzyme bonds to start locations on DNA. Secondly, the DNA molecule is split. Lastly, RNA bases are added into the single helix structure. When they are added, they are always added the opposite of what DNA is in DNA. For example, if we have cytosine, it is changed to guanine. With thymine and adenine, it is a different story. When there is thymine, it is normally changed to adenine, but then when there is adenine, it's not changed to thymine. It is changed to uracil. They use uracil as an RNA, there is no thymine. Now every 3 bases make up 1 codon. These codons are used in a process called translation. And in translation, they take every codon and see what code it's for. Every codon is a specific code for an amino acid. There are stop codons and there are start codons, which are codons that tell the RNA where to stop and where to start. These amino acids that are coded by the codons then bond together to make proteins. During this process, mutations can occur. These mutations occur because DNA replication is not 100% accurate all the time and sometimes makes mistakes. There are different kinds of mutations. Mutations are changes in the DNA. Mutations are not always tremendous changes to your body. When mutations form over time, they are likely to be much weaker than inherited ones as they form in one cell. When mutations occur from an inheritance, it is more drastic, but not necessarily negative. They are more effective because these mutations start out in the sperm and egg and then develop in the individual. This is how mutations can affect a big quantity of cells. They start to affect more proteins, which affect physical features of that individual because proteins can affect the look of your skin, nails, hair, and more.